Thank you so much, Jobril. What an interesting talk. We look forward to the interesting questions that will come after this. No, so I'm sure that you can, or I hope that you can stick around to answer the questions. Okay, thank you so much, Jobril. What an interesting talk. We look forward to the interesting questions that will come after this. No, so okay, so I guess we have to call on the Q&A facilitator, and this is Dr. Larry Ann Ipulan to start with the question and answer session. Larry, I guess we have to.
Yes, Ma'am Sonia. Hello. Thank you, Dr. Pinto. And thank you, Dr. Pelayo and Job, Sir Job, for your presentation.
Thank you, Ma'am Larry.
Si Job kasi yung presentation niya, it's part of the program where we are together, co-projects in the same program. So it's very interesting for me too. And then also, of course, Dr. Pelayos, asan na ba si Dr. Pelayo? Andito pa.
Hi, ma'am. Thank you po. So, thank you, everyone.
Thank you for giving some of your time, sharing your time for us. So, ano ka dyan?
Thank you, ma'am.
Wala ka ba sa lab ngayon? Are you in the lab?
Wala, ma'am.
Okay.
No, ma'am.
Okay. Okay, so I was just going to read again. So if anyone else are interested to send your questions, so please use the Slido link na nasa chat, or you can use this QR code, scan this QR code, and then in the Slido, you can actually change your name, and then just write the name, affiliation, and then your question. Or if you want to ask a question anonymously, then you may do so, and then just type your questions, and then send it in to Slido. Okay, so let's start with a question muna. So 2 questions first for Dr. Pelayo. So first question is, thank you for the interesting talk, Dr. Pelayo. What is the practical application or use of your research on biotimer genes and biotimer regulation?
I think we're thinking po na for agricultural applications po, so hopefully what we have po, like manipulating knuckles expression, timing, if applicable po to other genes, it's one way po to regulate gene expression maybe in other crops po, and hopefully it can contribute later on to our food security. It's a bit later, medyo later pa siya na application. We're still lots of things to understand and to try to find out, but hopefully we can help with what we're doing po. We can somehow contribute to that po. So yeah.
Yes, that's timing. Parang timing ng floral differentiation.
Flower development.
Yeah.
For later on, organ formation sa fruits or greens po. And we think na the biotimer, maybe it's also coordinating the environmental responses po. So maybe with abiotic stress po. So yeah, for climate change related responses for that.
Okay. Thank you for your answer, Dr. Pelayo. Actually, my question ako sa iyo. Because I find your this talk you had. So, ang question ko is, the delay of KNU induction, does it result to delay in floral? Does it mean that it will result into delay in floral differentiation? So siguro, medyo I'm curious about the main phenotype doon sa main phenotype change in the KNU.
For our mutants, we didn't use what we call the rescue line. So intact pa rin po yung endogenous knuckles dun sa system. So we didn't really see like drastic phenotype changes were related to our mutant. Pero for yung knuckles din po, kaya yung name niya ay knuckles, they found out sometime before na if they knock out the knuckles a gene, it would result in like carpal a strange carpal development and looks like knuckles, so it got the knuckles name. So maybe if the endogenous knuckles was knocked out and then we put our gene there, maybe we will see phenotypes related to like also carpal development. Maybe hopefully yes, about the timing also for development later on. But yeah, we didn't really use rescue lines to show that po. So yeah.
Thank you, Dr. Pelayo. Sige, pahinga ka muna konti. Let's have a couple of questions naman for Job. Okay, Job. Question from Dr. Nadia from Silliman University. Does the DENR have standards for EDCs in water? If not, how were the EDC values you detected in local water samples compared to US EPA standards?
Okay. So thank you po, Ma'am Larry, for the question. With regards po to the DNR, there's still no particular regulation or framework at hand. In fact, we're actually proposing on working on an initiative to propose these frameworks, because as of the moment, DNR cannot really prepare these guidelines, because in the first place, there's no basis or baseline threshold guidelines that can be used. So there are no studies that show the presence of EDCs whether it is strong enough to actually support a regulation. So as of the moment, we still need to apply this. Now for the particular regulation, so the US EPA actually declared this particular guidance threshold based on their it's actually more of based on their comprehensive outline of all the publications, wherein they found that if a particular chemical reaches a level that is beyond, let's say an estrogenic substance reaches a level beyond that, there are consequences in the cells and also in the organism. So most of these regulations are actually very arbitrary as of the moment, because they're still trying to figure out, should we reduce it or should we increase the level, the guidance threshold. So as of now, the most recent published is 0.7 nanograms. But eventually, we can actually devise the method to change its guidance threshold and adjust the guidance limit by just changing the positive controls.
Okay, thank you, Job. Oo nga, wala pa for EDCs, wala pa talaga.
Wala
under DNR, right? That's right. Okay, and another question. Okay, so knowing what you know about EDCs in local water samples, would you recommend that people avoid drinking from the tap, especially in Manila?
Actually, not necessarily, because we still need to confirm this first pa. We have data that says that there are levels that are reaching the guidance threshold, but there are also instances wherein the water may also be in a bad situation because for example, a storm went in and then that caused the site or the ground where the water is coming through to become disturbed, and this increased the flow of certain contaminants from that particular treatment facility. And also another thing to consider is whether the treatment facility is also having problems with their treatment process, because as shown in one publication, the treatment facilities can actually help reduce the EDCs in their water samples. So it's just a matter of monitoring and surveillance to make sure that water will retain the levels that are accessible to the guidelines. So for now, just continue drinking. It's definitely safe. We just need to confirm further if that level is consistent.
Okay. Siguro in relation na, no, to your answer, because there's a third question here. So the question is, after detection, how are EDCs generally extracted or removed from bodies of water?
So for the extraction process, there's still a lot to. It depends actually on what type of EDC you're working on. Some methods are more persistent and more difficult to extract, like PFAS, because they are very what you call this? They are known as the they don't necessarily degrade that easily, so they can stay there indefinitely and they will not be degraded. But there are also EDCs that can easily be mitigated by actually maintaining the water and ensuring that there are no plastic bottles, waste contaminants, or effluent that are flowing to the system. Eventually, they will naturally degrade, and then it will reach a stable level. So it depends on the particular EDC.
So there are different ways, but we
Opo.
don't have that yet kasi hindi pa nga
As of the
ginagamit
moment.
ng yes, because the
Opo.
DNR, they're not aware of, they don't have regulatory parang policies for the EDCs.
Mm-mm.
But hopefully your research will contribute to the push of that policy. Siyempre, kailangan mo nang ma-detect bago
Opo.
muna ng pansin yung paano siya tatanggalin, right?
Yes.
And so another question naman kay Dr. Pelayo again. So, Dr. Pelayo, is the biotimer mechanism independent of the effects of the plant circadian clock oscillators? Or does the circadian clock influence this process?
Currently, we haven't checked for the influence of circadian rhythms in our biotimer mechanism, but right now, we're saying po na it's intrinsic and maybe a bit separate from circadian rhythms kasi from how I understand circadian rhythms, very little. Circadian is a bit more about like the diurnal cycles and how plants or organisms would sense the like daytime or nighttime time. So right now we haven't really checked how our biotimer is um related to that. But yes, yes, they're possibly linked, but right now po we haven't clarified any connection yet, so can't really say definitively po. But yeah,
Yes.
thank you for the question.
Oo nga, very interesting din. Although yung meristem, floral meristem development, they are different each bud, right? They are timed differently. Tama ba? So,
Yes, ma'am.
so in that case, pero may ano na ako pahabol na question lang din about, so kung nare-reduce or nade-delay yung induction pala ng KNU, ng knuckles, does it mean na continuous yung proliferation?
In this case, kasi ma'am, like for knuckles, it's transitioning na from proliferation to differentiation. So, magsa-stop na rin po yung mag-stop na rin ang proliferation. Yes, good question, ma'am. Kung nade-delay din po ba yung proliferation niya. We also haven't really checked kasi we haven't, like, we weren't able to do experiments that like synchronized ourselves, and yeah, it's still that part is also still a challenge. Yeah, the cell cycle bits are still yeah, still I was still trying to optimize kasi I went home na po from Japan, so up until the last minute I was still trying to optimize my cell cycle experiments. So yeah, it's still, those parts are still remains a challenge, but yes, it would be really nice to be able to demonstrate that connection if the proliferation is also extended. So yeah, yes, very very good question. Thank
So
you.
hopefully, we will hear more from you along this
Yes,
topic. Before we go back to Job, so last na lang, Dr. Pelayo, if you have any message for our, we invited kasi, one of our board members is a high school teacher, STEM teacher. So if you have any short message for the students, whether undergraduate college students or mga high school students who are interested or budding scientists at this moment. So do you have any message for them?
Oh wow. Yeah. Thank you. Oh wow. For the wedding science. Yeah. Um. Uh, I hope that our talks today, also Sir Jobriel's a very nice talk also. I hope that it encouraged you guys to keep pursuing your interest in science and the sciences. Um Even now, for me, it's not an easy path. I'm still I'm still trying to find my way. So if it's that sort of life, it's not like a glamorous life, but we keep learning and we keep growing. And you are if that sort of life or like endeavor appeals to you, I hope that you will also um pursuing it. It's not an easy path. Experiments fail all the time. Like 80 to 90% of the experiments fail, and what you see on the slides are like the 10 10% that work. But yeah, but yeah, there are joys to it too. And yeah, not that I'm trying to discourage, but like yeah, maybe what I'm doing is the opposite. But yeah, but yeah, no, I admire you. I admire the students, the young people who are into science. And yes, I hope that also through these talks, I think our roles now as like the presenters or like the teachers is to try to nurture the younger students. And we hope that we're also going to do our best to for those who are interested in pursuing the scientific path. We hope that we can help you on our way too. So yeah, if you're also interested, please feel free to contact us and also to participating. And yeah, thank you so much for listen thank you so much for listening to our talk. So yeah. Yeah.
Thank you, Dr.
Thank
Pelayo.
you.
Yes, mahirap pero exciting naman, di ba? It's not that
Yes.
glamorous, but it's really exciting.
Yeah.
And then, so let's proceed with Job naman. So Job, actually, napag-usapan kasi may question about the drinking water. You also included in your research to test iba yung mineral water and distilled
Yes po.
water.
Opo.
So, how did it fare with, in comparison with the level ba that?
um well, fortunately they passed. We're working on like making sure that let's hope that all of them always passes, like the mineral water and the drinking water, because they'll be my basis, or they're like my controls to make sure that, well, this should be the closest thing that we can hopefully achieve in our drinking water. But let's make sure that we keep it that way, and then beyond that we may not we may want to reconsider our approaches in maintaining the water quality.
That's
So
right.
fortunately they passed.
Okay. And hopefully they keep on passing. And if
Yes.
not, you will be there.
Yes.
Monitor them.
We'll keep
Okay.
our surveillance on.
Yes, yes. So, and also, same with Dr. Pelayo. I know that you're still one of the young scientists here, but Dr. Pelayo looks very young also. So, do you have any message to our
uh,
budding scientists?
um, na pressure ako sa first answer ni Ma'am Pelayo. um I guess if you're passionate about science, you keep pursuing it, and then if you start to question why are you pursuing it, then you go back to why you did it in the first place, siguro. So are you passionate? Are you still passionate with it? If not, it's okay. You can move forward to a different path. Pero if you are still interested in that field, then do consider pursuing it, naman. And I guess this is the reason why we open these talks, these conferences, is to make sure that everyone is aware that this is what can make or break like the passion for your study or for your field, and it may also become the source of like a spark for the students to actually pursue the science and maybe to generate like a path for them. So yun lang. So be passionate and maintain that grit lang siguro if you are into science.
That's right. Okay, so just like everyone of us here na nag-start na nanood ng mga webinars or
Yes.
mga seminars, we want to contribute to the body of knowledge. It's basically our, ano naman, diba? Our goal as scientists. So if you want to be one of us, clickish, if you want to be one of us, no, you can. If you choose to do so, kailangan lang talagang perseverance, right? Okay, so,
Yes.
and you have mentors like Dr. Pelayo and Job na rin. So, there are more and more people are entering science. So, please, just know the right people to reach out to. And especially PSDB, if you're interested, we can connect you with those that are doing developmental biology research work. And through our conventions and itong mga webinar series, we can introduce you to more and more scientists. Ayan. So thank you again, Dr. Pelayo and Mr. Baluyot. And to show our appreciation, next slide, please. We would like to give this certificate of appreciation to Margaret Ann F. Palayo, PhD, and next slide,
Thank you.
and Jobriel C. Baluyot, MSc, for the generous sharing of knowledge and expertise as resource speaker during the PSDB 2022 webinar series session 2, held online on April 30, 2022. Signed by our program chair, John Vincent Navalan, another young scientist, Dino T. Tordesillas, PhD, youngish, no, young, si Dr. Dido, and of course, yours truly, President Larry Annie Pulan Colette, PhD. Elder na
Thank you,
daw,
ma'am.
elder. Okay, so thank you very much, Job and Dr.
Thank
Pelayo.
you po.
Hopefully, maging member
Thank
kayo
you po.
and we can invite you again for another talk. Thank you. So that's it for my part, Dr. Sonia.
Yes. Next slide. Okay, for the next part, we would like to hear the closing message from our Vice President of PSDB, Dr. Nadia Palomar Abasamis.
Good afternoon to everyone here in Zoom and to those watching through our live streaming platforms. This year's webinar series was a showcase of innovations and biotechnology as applied to the fields of developmental biology, environmental science, and agriculture. On behalf of the members and officers of the Philippine Society for Developmental Biology and all the students, teachers, and professionals who joined us in the past 2 Saturdays, I would like to extend our sincere thanks to our speakers, Dr. Lilia Fernando, Dr. Euphrosyna Atabay, Dr. Margaret Pelayo, and Mr. Jobriel Baluyot for sharing with us cutting-edge knowledge, techniques, and applications associated with or relevant to animal and plant development. It is always great to see science and technology being used for the improvement of society. Mabuhay kayong lahat. To all our participants, we hope that this year's webinar series was an enlightening experience, and we hope that it has stirred your research interests or maybe even inspired some of you to follow the career paths of one of our speakers. Thank you for your support, and we look forward to seeing you in PSDB's future activities. Take note, this includes our masterclass series and the 14th Annual Scientific Conference of PSDB this coming November. Once again, thank you very much and have a pleasant weekend ahead.
Thank you, Nadia, Dr. Palomar Abesamis. I guess the next part is the photo session. May I call on Mr. Jordan Halili to give the rules for the photo session. Jordan, muted ka.
Nakamute. Alright, pwede na ba natin i-turn on yung mga cameras natin? Meron po tayong background na pwede nyo gamitin. And, yan. Sige po. Check ko lang po yung gallery. Yan. So, let's give time for others para i-on yung camera sila, mag-ayos ng buhok at magpa-fresh para ready sa picture. Okay? Okay. So, i-close ko lang ito mga close. Yan. Alright. So in total, we have 3 panels. So sige, meron pang mga nagbubukas ng camera, humahabol. Sige, hintayin po namin kayo. Ayan. Kitang-kita ko yung mga nag-aayos ng buhok ko. Nagre-ready sila eh. Okay. Sige, I think we can start na. Okay, for the first panel, so medyo i-hold lang po natin yung smile natin for a while kasi hindi ko sure kung nasang panel kayo. Pero sige, let's start. For the first photo sa first panel, we have 3, 2, 1. Ako pala hindi nakatingin ulit. 3, 2, 1. Okay, next panel. 3, 2, 1. And then for the last panel, 3, 2, 1. Alright. Thank you so much po sa lahat ng mga nag-participate. Ma'am Sonia, back to you po.
Okay, thank you, Jordan. Okay, so I guess let me just repeat on how one can get a certificate of participation. You must be an official member of PSDB, registered the PSDB 2022 webinar series, and you must submit the evaluation form for each session. And you must take note that the evaluation forms will open at the end of the session, end of this session, until 11:59 p.m. today. So lastly, we cannot have made this far if not for our very reliable and sponsors who are always with us in our webinar series and other activities: Macare, Merck, MedTest, Afil-Ana Serve Incorporated. We express our deep appreciation for your presence and your support. Thank you so very much. And to everyone, thank you for your participation as we look forward also to your presence in our next activities. Just like Dr. Palomar Bisames said, this includes our master classes and also our annual convention nearing the end of the year. So thank you so very much. And well, this is not goodbye, but maybe just a little bit of farewell, a little, and then see you again.
Thank you po.
Thank you, Ma'am Sonia.
Thank you, Job. Thank you, Dr. Pelayo. Thank you for all our attendees.
Thank you sa lahat.
Thank you po.
Yung evaluation form po, kindly scan this QR code. Or click the link sent by Dr. Nadia. Thank you, Ma'am Nadia.
JV,
Yes, ma'am. Hello po.
kita mo yung message ko sa'yo. Nag-direct message ako.
Yes, ma'am. Opo. Dapat po per session.
Oo nga. So,
Papakorek ko na lang
i-edit.
po dun.
I-edit lang yung
PSDB
form.
page.
Baka matakot yung mga tao.
Okay po. Sir, di po
Okay.
yata sa part